gave you an overview of some of the things that I'm going to be talking today. So we're going to go over some of the same things and then some new things in more detail. So the family, so the Rho GTPases is a family of genes. So there's 23 genes in mammals, and it can be further subdivided in different subfamilies. The ones that I talked about yesterday included the best characterized one, which includes the RAC subfamily, the CDC42, and then the Rho A subfamily. So these proteins are small proteins, around 21 kilodaltons, and they are conserved from yeast to mammals, and they're highly homologous. So they're about, if you align all the GTPases, they're about 55% homologous, 50 to 55% homologous. But between subfamilies, they're very, very closely related, 90% in many cases. So humans have 23 genes. There's 7 genes in yeast, 11 genes in plants, and then the C. elegans has 9 genes, and Drosophila has another 9 genes. Most of the best characterized, sort of the key regulator like Rho, RAC, and CDC42 are conserved in most organisms, with the exception of yeast that doesn't have RAC. So this family can also be subdivided in what's called the classical Rho GTPases, which are shaded in blue, and include the sort of the best known ones, Rho A, RAC, and CDC42, and then the atypical ones. Some of the characteristics that make these proteins atypical is that they are usually not regulated in the same way as the classic ones. And that, for example, some of these proteins, including, for example, the round subfamily, they're always in the active state. They're always in the GTP-bound state, so they don't switch between GTP and GTP. And they're usually regulated in different ways. For example, they can be regulated by phosphorylation or by targeting to different subcellular sensations. So we're going to go over the cycle again, so I apologize if some of the slides are repeated, but I think that they're important sort of to keep the flow of the talk. But if you remember from yesterday, Rho GTPases are described as molecular switches. So they can be on or off, and that's basically what the switch means. So, and the on and off state is defined by their ability to bind a nucleotide, in this case GTP, which defines the activated state, and then GDP, which defines the inactive state. So the switch between these two states is very, very slow. And it occurs at a very slow speed by itself. So it's usually catalyzed by 2 families of protein. Okay. So when the proteins are in the active state, they can bind to a series of proteins called effectors that bind exclusively to the GTP-bound state, and then they usually catalyze different signaling pathways. Most of the effectors are kinases, but there are also some other type of proteins that bind to the GTP-bound form of GTPases. As I said before, there's 2 families of proteins that regulate this exchange. The one in nucleotide exchange factors promote the activations of the GTPases, and I'm going to go over in detail of this family of proteins. And then the GTPase activating proteins, or GAPs, what they do is they promote the hydrolysis of the GTP into GDP, and that's the inactivation. So again, as I showed yesterday, a 3rd layer of regulation is done by the Rho GDI family of proteins. These proteins are important to sequester the inactive pool of GTPases in the cytosol. They're also important for targeting to membranes and extracting from membranes. So what are the functions of these proteins? These proteins are all over the place. So there's hardly a process in the cell in which you cannot find a Rho GTPase that's involved. For example, cell adhesion, the adhesion to the extracellular matrix, formation of focal adhesion, development of stress fibers. It's all regulated by the Rho families of GTPases. The development of contractility, for example, smooth muscle cells in blood vessels. This is regulated by Rho GTPases. Migration and invasion, these are behaviors that can occur during development. During embryogenesis, you need particular types of cells to migrate and move to other parts of the embryo. They also occur when these processes are not regulated during metastasis in cancer cells. They migrate when they shouldn't be migrated, and they move to and invade to different tissues. This is also regulated by GTPases. Cytokinesis at the end of mitosis, when the cells need to divide into daughter cells. The formation of the contractile ring, it's a process that is regulated by Rho GTPases. So when there's defects in this formation, in this process, you can have multinucleated cells or defects in the completion of mitosis. The development of cell polarity. This is a very important process that occurs on all epithelial cells, in which you have two types of membranes, the apical membrane, the basolateral membrane. A lot of it is regulated in particular by CDC42. Another type of polarity is a polarity that you can see in an axon, where you have in a neuron where you have an axon and a dendrite, and it's very similar to what happens in epithelial cells. And again, Rho GTPases play a very important role in the development of polarity. Other processes that we tend not to think about, like the internalization of fluid components from the extracellular environment, like macropinocytosis, internalization of bacteria, or other processes like during inflammation, when leukocytes need to extravasate out of the bloodstream into the inflammation site, are also regulated by Rho GTPase. So I showed you this yesterday, and this was sort of the experiment that started the whole thing. So this is from a review in 1998, but in the early 90s, around 1992, a couple of groups, they were mostly studying RAS GTPases, which shares a lot of similarities with the Rho GTPases. And they found these proteins, which initially were named P21 activating GTPases, later named Rho GTPases, and they started doing experiments to see what these proteins were doing. And the way they went about it, and this is an era before transfection, before siRNA, and before a lot of things that we take for granted today. So the way to try to see how these proteins worked was to purify them and then microinject them into cells. So they had the purified, for example, Rho protein, and then they would microinject it in the single cell, and then they will stain for actin, stain for other markers like focal adhesion markers, and just look to see what was the phenotype that was induced by having higher levels of higher activity levels of a particular GTPase. And what they saw was when you look at Rho, so they tested the three major ones, Rho A, RAP1, and CDC42. What they saw when they look at Rho A activation was that compared to resting cell, which is this panel, when they put Rho A in the system, they have an increase in the number of stress fiber formed, and these are actin-rich bundles that can promote contractility. And then they also see an increase in the number of focal adhesions. What they saw when they induced RAC activation was that the cells spread more. They had a bigger area, and it promoted the polymerization of actin at the leading edge of the cell. So this is actin. This is again a focal adhesion marker. Instead of having these big focal adhesions that you see here, what they have is very small focal, what's now called focal contacts at just the very edge of the cell. And then if you look at what happens when you overexpress CC42 or you induce the activity of CC42, it's the formation of these very, very defined finger-like protrusions that are called philopodia that extend and are also actin rich and extend out of the cell. And these structures are believed to function in sensing the extracellular environment and also in adhesion. So just remember that these phenotypes came from these cells. So this is the same set of cells that were transformed with different GTPases. So the changes in the phenotype are quite dramatic. So if you look at these proteins a little bit more carefully, so one of the things that is important about them, and I show that in the cycle, is that when they are active, they are in the membrane. I talked about that yesterday in some detail. And if they are not in the plasma membrane, usually their effect is not transduced. So basically there's no signaling that occurs if these proteins are not in the membrane. And one of the reasons that these proteins can be targeted to the membranes is the presence of a lipid group at the C-terminus. So as you can see here, and these are all the GTPases or all the major ones, and as you can see here, there's different ways that these proteins can be modified. So what they have is a sequence called a CAX box, and CAX means a cysteine and then 2 amino acids that are usually hydrophobic, and then the last amino acid could be anyone. So there's two ways that the Rho GTPases can be modified with two different lipids, a 20 carbon lipid and a 15 carbon lipid. The 20 carbon lipid is called geranyl, and it's here depicted as a GG. And then other proteins can be farnesylated. This is a 15 carbon lipid. And then there's special cases in which these proteins can have two different lipids, either a geranyl or a farnesyl, and this is the case of Rho B, and another lipid because they have an extra cysteine that's called a palmitoidal. So these proteins are going to be more tightly associated with membranes than the ones that have a single lipid. So this is very important. If you don't have the lipid, the proteins cannot perform their function. So how do they get the lipid? I showed this yesterday, but we can go over it again. What happens is proteins get synthesized in the cytosol. They have this sequence, the CAX box. Again, the CAX box, an enzyme, either the geranyl transferase or for farnesyl, the farnesyl transferase, can add the lipid to the cysteine, and then the protein is transferred to the endoplasmic reticulum. It gets further modified by cleavage of the last 3 amino acids, and then methylation of the last amino acid. And then after it's mature, we believe that the GDI will come and take it out to the cytosol and shuttle it to different membranes in the cell. So the GTPases have several particular structural elements. So in particular, and I'm going to rotate this structure in a minute, but you can see here that you have, this is the nucleotide, so it could be GTP or GDP. And then in blue, there's just a fragment of an effector. So this is an effector binding to the GTPases. So there's two very important structural elements on the GTPases. They're called switch 1 and switch 2. And these are the elements that are important for the binding interface. They're important for binding not only the effectors, but only the GAPs, the GEFs, and also the GDIs. So it seems that everything that binds to the GTPase binds in the same place. So you cannot bind an effector and a GAP at the same time. You cannot bind a GEF and a GAP, or a GEF of an effector. So there's only one thing that can be bound on a GTPase at any given time. So let me play this movie so you can see how. So if you look at the blue structure, which is the effector, you can see that there's it interacts. The surface of interaction overlaps with where the switch 1 is and the switch 2 is. So this is the surface of the amino acids that are most important for the interaction. And mutations in these amino acids can prevent binding to GAP, GEF, and effectors. So this is surprisingly, when people crystallize these proteins, they crystallize them both in the GDP and the GTP state. They found that there's not a big conformational change. Conformational change is very small, but somehow it's big enough that it allows something to bind that it couldn't bind before. So other GTPases of the same big family, the RAS superfamily, like ARF, have a huge conformational change when they bind to GTP. The Rho GTPases, not so much. So this is a busy figure, but I'm trying to walk you through it. So this is the Rho GTPases, and this is from a paper of Dvorsky in 2004. What this investigator did is they aligned, they compiled, there's about 20 crystal structures between GTPases and GAP, GEFs, and effector. So what they did is analyze all these structures, and they compiled what amino acids are interacting with the particular interacting proteins, and they put it together as a frequency graph here. So what you can see here is the structures that I mentioned, the switch 1, the switch 2, the prenyl group, and some other important structures. And what you can see that the amino acids that interact with GDI, exchange factor, GAPs, and effectors are mostly in the same region. So if you look at, for example, switch 1 and switch 2, this is where most of the things happen. So if you look at GDI, it will interact with amino acids here, here, and maybe some other interactions, especially the prenyl group. If you look at GEFs, same thing, mostly with switch 1 and switch 2 amino acids. Same thing for GAPs, same thing for effectors. There's a couple of differences that you can pick up here. You can see that GAPs and effector bind to this other structure called the P-loop. And you can also see that, for example, GDI interacts with region, which is an alpha-3, a helix named alpha-3. And also, as you saw from the structure yesterday, and I'm going to show you today again, it interacts very, very efficiently with the lipid domain. But other than that, it's very, very conserved, the region of amino acids that mediate the interaction. Okay, so I'm going to talk now a little bit about the Rho GAPs. So how are the GTPases activated? So I mentioned that they need to exchange, the GDP needs to be released, and the GDP needs to come in. So the affinity for, so there's a couple of things that are important when we think about the exchange reactions. First, the affinity of the Rho GTPases for both nucleotides is very, very high. So one could think in a sort of a simplistic view that the GTPases have a very high affinity for GTP, very low affinity for GDP, and that may help the exchange, but that's not the case. If you give them GDP or GTP, they will bind with equal affinity. This is not how it happens. So having such a high affinity means, of course, that the dissociation rate is very, very low. So if you leave a GTPase in solution and you wait until the GTP is kicked out or the GDP is kicked out, it will take sometimes more than an hour. So this is not how cells work. In cells, these things happen in the order of seconds or in the order of 1 minute. So if you can activate a GTPase very, very fast, almost instantly. So this is not the, again, this is not the way it's going to work in the cell. And this is what I just mentioned. The exchange in vivo occurs very, very fast. And it has been shown that the exchange factor accelerates the reaction by several orders of magnitude. So if you have it with or without the exchange factor, this is going to make it a lot faster. And the other thing that we're going to talk a little bit about is that the exchange factor are parts of the nodes, the targets of the biological signals that it will induce or inhibit their activity. So there's a lot of regulation of these signaling pathways that happen at the level of the exchange factors. So how does this work? So there's two series of reactions. First is the binding of the exchange factor and the dissociation of the nucleotide, and then is the binding of the GTP and the release of the exchange factor. So I'm going to show you sort of a cartoon, a cartoon rendition of how this works. So let's say you have a GTPase, and this yellow molecule is a GDP, the inactive molecule. This is the base and the phosphate group. So you have the GTPase bound in the inactive state. When the GTPase is in the inactive state, the GEF recognizes it as a substrate. So the GEF will associate at the beginning through a weak interaction with the GTPase in the inactive state. What would then happen is that the GEF destabilizes the nucleotide binding pocket and will just start releasing the nucleotide. And this still a weak interaction, but at some point the destabilization will prevent the nucleotide from binding, and the nucleotide is going to be released. And this is what's called a nucleotide free. And if you remember yesterday, this is the principle that we use to identify or pull down exchange factor from the cells by making a mutant that is in this state, in the nucleotide-free state, because this is a very high affinity interaction. So what happens with the exchange factor promotes the release of the nucleotide. And again, this is a GDP nucleotide. So at this point, I told you that the affinity for GDP and GTP is the same. At this point now, the GTPase can bind a nucleotide, so it's open to bind another nucleotide. And it could bind either GDP or GTP with the same probability. The only reason that it binds GTP is that the concentration of GTP in the cell is about 10 times higher than the concentration of GDP in the cell. So probabilistically, what will happen is most likely now a GTP is going to associate with the exchange factor. And then, so first you're going to bind the base, then you can bind the phosphate. This is going to destabilize the exchange factor now, because the exchange factor does not bind to the GTP bound folder of the GTPases, and then the exchange factor will be released. In theory, there's a possibility that another GDP would have bound, and then you go through the cycle again until at some point you get the GTP bound and release the exchange factor. So this is the Rho GAP family, and I think that I gave you this review to read. It's an extremely complicated family. It's about 69 genes in the human genome, and as you can see, it's extremely diverse. So the only thing in common of all these proteins is these yellow and blue domains. There are the DH and pH domain. This is what represents the catalytic domain, and that's what this family is called the Divel homology subfamily. Divel homology is this is what stands for DH, and Divel is the first exchange factor that was characterized. That's why after that everyone was Divel homology, something related to Divel. So one of the things that you can see when you look at this is the diversity of the protein domains that are present in the different exchange factors. This is what people think, although there's not a whole lot known about this. This is what determines when and where they work and how they're activated. The ability to bind to other proteins, the ability to get phosphorylated, the ability to get targeted to different parts of the cell. And then once they can activate the GTPase that they are specific for. The other thing that this graph shows in these little dots here is what GTPases these exchange factors are specific for. If you can see here, Rho B, Rho C, RAC 1, 2, 3, etc. You can see that, for example, this one is Rho A specific, but then some others, like this one, are specific for all the GTPases. So that's another important thing when we think about regulation of Rho GAPs. They can be highly specific for just one GTPase. They can be highly promiscuous for all the GTPases. All right, so I mentioned this. So there's usually what characterizes this family is a DH domain. This is the catalytic domain per se, and then it's most of the time followed by a pH domain. It's called a plexstring homology domain. And then, of course, the DH domain is a catalytic domain. The pH domain has a lot of functions. In many cases, it can bind lipids, and this is believed to assist it in targeting these GEFs to membranes. But also, in a few cases, has been shown to assist in the exchange. So the catalytic reaction occurs a lot better if you have the DH-pH than if you have only the pH alone. And it's still not well understood how this happens, but it's very clear that in many cases the pH domain is essential for the catalytic reaction. And then I mentioned this multiple protein-protein interactions domain, and I'm going to come back to that in a few slides. And then the single and multiple specificity. And that's also something that I'm going to talk about later. All right, so for a few years, people were happy with the DH-pH family, and then they started finding these other proteins that were able to exchange and activate GTPases, but they didn't have a DH-pH domain. So there's another 11 members now in the family of exchange factor. They call ZZH subfamily, and they come in two flavors. They're called the ones that are related to this protein, which was the first identified, the 180. Another ones are related to zizimine. They, in essence, catalyze the same reaction, release of the GDP, rebind of the GTP, but the catalytic domain is absolutely not conserved. It has nothing to do. So these proteins most likely evolved independently to perform the same function. So there's two sets. So this family can be subdivided again now in two subfamilies, as I said, and they have 11 genes in humans, and there's typically, I mean, there hasn't been one found to exchange on Rho. So they're usually RAC specific or CDC specific, and the DOG180 are usually the ones that exchange on RAC. The zizimine are usually the ones that exchange on CDC42. So they're a little bit more specialized than the DH-pH subfamily. They have these two domains called CCH1 and 2, which are these two that are important for the exchange activity. And as I said, they can be subdivided in these two classes. But they're also conserved even in early eukaryotes. So again, like the DH-pH domain, this is a family that it's evolutionary old. So how the Rho GAPs regulated? So a simple way to think about it, and initially people thought about it that way, was, okay, in different tissues we have different exchange factors, and then they're going to be related in different ways. We may have some in brain, some in liver, some in kidney, or something like that. It turned out to be that that was not the case. So most of the exchange factors are expressed in every tissue. So there's a couple of them that are specific for brain. There's a couple of them that are enriched in different tissues. But this is in any way something that can explain why you have so many. In any given cell, at any given time, you probably have 40 or 50 or maybe more exchange factors. So one of the first clues about how these proteins were regulated came from studies in which they found that truncations in the N-terminal part of the exchange factors will render them constitutively active. So that was a clue to think maybe when these proteins are full length, they are somehow folded in a particular way that prevents the activity from happening. And then through post-translational modifications like phosphorylation or some other events, these proteins now open up, can exchange and activate the GTPases, and then at some point they get inactivated again. And there's a lot of evidence for that. I'm going to show one or two examples. There's another couple of examples that show that these proteins can be sequestered in different parts of the cells, in particular in the nucleus. And I showed a little bit of that yesterday in the exchange factor that I talked about, NE1. So there's a potential of a protein being targeted to a particular protein cellular compartment, and then at some point released from that compartment, performed in function, and then get activated again. And then there's a lot of mostly circumstantial evidence, but there's some publications showing that there's proteins that will bring the exchange factors to a particular part of the cell together with other components with the same pathway, including receptors and including downstream effectors. And that could be a way to compartmentalize a signal within the cell and to use a particular set of molecules in one pathway and then other particular set of molecules in another pathway. And I'll show a couple of examples of that too. So one of the most studied exchange factors is a VAB family of proteins. So 3 genes, VAB1, 2, and 3. And in this, so this is sort of the best example of intramolecular inhibition. So this protein has many different domains, a couple of protein-protein interactions, domain SH3 domain, SH2 domains, this domain called C1, and then the catalytic domain, and 2 other domains, a calponic homology domain and what's called an acidic domain. In this acidic domain, the 3 tyrosines. These tyrosines have been shown to be phosphorylated. So this protein is folded on in itself, and it's inactive when it's not phosphorylated. And there's a couple of different layers of regulation of this protein. One is phosphorylation. So when this protein gets phosphorylated, this interaction between the pH domain and the acidic domain weakens, and this protein sort of open up. When this protein is now phosphorylated, there's other proteins that can interact with the CH domain, sort of stabilizing this conformation of the protein, and then also lipids like PIP3 can bind to the pH domain, further stabilizing this conformation. Now the GTPase can bind, and it can get activated. And again, when the protein gets desphosphorylated, this goes back to this state, and it gets inactivated. VAB proteins are regulated by a lot of different pathways, but mostly receptor tyrosine kinases. Things like PDGF or EGF will activate the receptor tyrosine kinases that can phosphorylate these residues, and then they can promote the opening up and activation of this exchange factor. So I mentioned subcellular sequestration. One of the examples that I told you is a protein can be sequestered in the nucleus, and then somehow a signal can get them out of the nucleus and activate it. There's one of the best studies examples does not include the nucleus, but includes a GEF that can bind to microtubules, and this is a protein called GEF-H1. GEF-H1 is a mostly Rho A specific GEF, so it will activate Rho A. And what happens with this protein is that it can bind to polymerized microtubules through several different domains. So when it's bound to microtubules, it's inactive. It's just sitting there, and it cannot activate Rho A. When there's an event that promotes the depolymerization of microtubules, this protein gets released, and it's now active, and it can now activate Rho A. And there's a very simple way to test this, and this is how people realize that this was happening. Is a lot of researchers in the lab use drugs that depolymerize microtubules for their research, and one of the best known is nocorazole. What nocorazole will do is very, very fast. If you treat cells with this drug, all the microtubules will get depolymerized. What people saw when you did that is that the cells that were treated with nocorazole had a lot more stress fibers. So that's when they realized that Rho A was being activated, and that's when they started looking for the responsible for this change. and they found that GEF-H1 was that. And I'll have one little example here. This is a cell, a control cell. This is a cell that was treated with no cortisol, and as you can see, there's a lot more stress fiber in this one than in this one. And it's very clear that something happened. And usually when you see more stress fibers, it's usually associated with a Rho H phenotype. So this is the sort of the second mechanism of regulation. So the third one is the scaffolding protein. This is a lot more complicated. I can involve a lot of different mechanisms. So it's basically an easy way to encompass all the rest of the mechanisms of regulation. And this is by far the least understood. But I wanted to show you this slide to try to represent what the problem is with this regulation of exchange factors. So if you look at the GTPases, I told you that you have around 20 GTPases in the human. And then the best known RORAC and CDC42. So if you look at the exchange factors, there's at least 3 times more exchange factors than GTPases. So already you have 3 times as many. But also, there's exchange factors that can activate more than one GTPase. And of course, one GTPase can be activated by several exchange factors. And then downstream of that, you have the effectors. There's at least 100 effectors in the cell. Many of them are specific and activated by a single GTPase. Some of them can activate it by more than one GTPase. So the problem is that at any given time in the cell, you have all these floating in the cytosol at any given time. So the question is, and this is something that I'm very interested in to understand in my lab, is what happens when you get a stimulus, for example, that activates this exchange factor. How does the cell decide which pathway to activate? How does the cell decide to go this way instead of going this other way? So this is one of the fundamental questions that people in the Rho GTPase biology are trying to understand. And one of the solutions is to what I mentioned before, is to have scaffolding proteins that segregate different components to different parts of the cell. So you can visualize this same sort of complex network in something a little bit more simple, in which you have different examples. So these sort of background shapes are scaffolding proteins that will bring together different components of different pathways. So for example, you can have here 2 receptors, the same receptor that can bind to 2 different scaffolding proteins. And this scaffolding protein will bind to a GEF, to the same GTPase, but maybe to a different effector. So then you can have one pathway signaling to one phenotype or one cellular response, another pathway signaling to another cellular response. Then you can have, for example, different scaffolding proteins that bind to the same GEF, but to a different GTPase. So then you can have an exchange factor that is able to activate RAC and CDC42, but depending on the context, with some receptor and effector activates a RAC pathway, with another set of components to activate another pathway. So this is a lot of this is theoretical, but there's a couple of good examples. So this is one example in which the same exchange factor. So these are 2 exchange factors, beta peaks and TM1. All of them, these 2 can activate RAC1. Beta peaks, when activates RAC1, in turn activate an effector called PAC. PAC can then promote actin polymerization and the formation of cell protrusions and induces cell migration, etc. TM1 can also do the same thing through activation of PAC. But also TM1 can bind to 2 different scaffolding proteins. And these scaffolding proteins will bring together different components of MAP kinase signaling pathway. So in one case, you get different MAP kinases, and that with JNK activation. Another one, you activate a P38 related pathway. So with the same GTPase, the same exchange factor, you can promote the activation of two different signaling pathways. And these are signaling pathways that end up with the activation of gene expression. So depending on which one you activate, you are just activating the expression of different genes. A very similar one, but related to Rho A. As I told you, Rho A and the Rho A exchange factors are usually associated with the formation of stress fibers, with adhesion and the development of contractility. And this is usually what happens if you activate this subfamily of exchange factor of P115. These are three very related exchange factors. They activate Rho, and they activate a signaling pathway that ends up in the polymerization of actin. But also, P115, again, combined to an exchange scaffolding protein called CNK1. This protein, CNK1, again, recruits some components of the MAP kinase pathway that are activated by active Rho, and then, of course, activate the expression of related genes. So again, you can, in a way, recycle or reuse similar components in different pathways by putting them together in contact with different members of a particular pathway. So I'm going to switch now to talk a little bit about Rho GAPs. So Rho GAPs are incredibly complex as the Rho GEFs, but also they're also really understudied in comparison with GEFs. If we don't know a lot about Rho GEFs, we know even less about Rho GAPs. So what we know is that the Rho GTPases are really bad GTPases. So they're really not a GTPase. Some people don't even like to call them GTPases because if you want them to act as a GTPase, they do it very, very poorly. So they do bind to the nucleotide, but they're very, very slow in hydrolyzing the nucleotide, which is what a GTPase does. So this is what it is. The intrinsic hydrolysis is really slow. It doesn't occur at physiological rate. If you leave a GTPase in the test tube, it will take a long time to hydrolyze the nucleotide. So again, in the cell, you need something else. And this something else are Rho GAPs. So the Rho GAPs interact with the Rho GTPases when they are in the GTP-bound state, and what they do is they promote the hydrolysis. And again, as for the GEFs, the rate of cleavage of the phosphates. So the GTP has 3 phosphates. What the Rho GAP will do is to promote the cleavage of the gamma phosphate, the phosphate that adds the last phosphate of the structure. It accelerates at least several orders of magnitude. So something that will take half an hour or an hour, it will take in the order of seconds or minutes. So how does this happen? So the way this happens is there's I guess for the chemistry fans, which I'm not, there's what's called a nucleophilic attack. There's a molecule of water that will coordinate with the last phosphate group, and then it'll promote the cleavage. And then this is what the end of the reaction will be. You have GDP, and you will release a phosphate molecule. The way it happens in the structure, again, the structure for the GAPs and the GTPases, several GTPases have been solved, and there's really good indication of how this works by looking at the structure. And this is my rendition of the structure, I guess. So you have the Rho GAP here. So the Rho GAP has one particular residue that is incredibly important for this exchange reaction, and it's an arginine residue. And then in the Rho GTPase, there's a very important residue, which is a glutamine residue, that also between these two residues, they coordinate both the phosphate of the GTP with the water molecule that I mentioned here, and then that promotes the nucleophilic attack and the release of the phosphate. So the fact that this glutamine here is so important and this arginine is that if you mutate any of these, then the reaction would not work. And this is a residue that is absolutely conserved, the glutamine. And if you remember, and you've probably seen in some of the papers that I gave you, the use of constitutively active or dominant negative is based on this structure. So the constitutively active mutants are usually glutamine mutations. So if you align all the GTPases, you can see that this glutamine is absolutely conserved. If you change this glutamine for a leucine, then these proteins are always in the GTP state because they can bind to GTP, but they're not able to hydrolyze it. They're missing this connection with the water molecule. So the mutants are usually called for Rho H Q63L and for RAC is Q61L. So that's the way it works. So this is the Rho GAP family. Again, an incredibly complicated family. And as I told you, there's still very, very much that is not known about how this family works. As for the GEFs, this is how they look. As for the GEFs, they have one domain that is conserved. This is what's called the Rho GAP domain here in red. All the proteins have it. You also have, similarly to the GEFs, GAPs that are specific for one GTPase. Here, this set of proteins are specific for RAC1. Then you have some of them that are specific for all the GTPases or for a particular subset of GTPases. So it's, again, very similar to what happens with the GEFs. So there's single specificity, there's multiple specificity. There's also, again, an incredible variety of protein-protein interaction domains that you can find in the Rho GAPs, again suggesting that the main regulatory events that control the function of these proteins are dependent on the presence of these different domains. So you can find protein-protein interaction domain. There's a lot of lipid binding domains in the GAPs. And there's some evidence that this lipid binding domain can also respond to things like membrane curvature. So when the cell changes its shape, then the GAPs can get activated or inactivated. But this is still a field that is in its infancy, and there's a lot that we need to learn about these proteins. So as I mentioned, the conserved catalytic domain, single multiple specificity, and then a wide variety of protein-protein interaction domains, and then there's a little bit more representation of lipid and membrane binding domains in the GAPs. So how are GAPs regulated? Again, there's very little known about it. But there's a lot of evidence on protein phosphorylation. There's also some evidence suggesting that binding to different lipids can promote their activation. Protein-protein interaction has also been described in a few cases, and then some other cases in which they show that a cleavage of a particular domain of the GAP can then get them activated. And I'll show a couple of examples. So P190 Rho GAP, this is a Rho A specific GAP. It's one of the best characterized. It functions in a lot of different processes like adhesion and migration and production of reactive oxygen species and many different processes in the cell. And it usually responds to both receptor tyrosine kinases, like things like EGF receptor or PDGF receptor, but also responds to the function of integrins, which are the receptors for the proteins in the extracellular matrix. So what happens when a receptor tyrosine kinase or an integrin get activated? What they do is they activate what's called a non-receptor tyrosine kinase, in this case SARC. SARC can phosphorylate P190 Rho GAP, and then that gets them activated, and it can now promote in this case, the inactivation of Rho A. So this is a very simple one, and one that makes in a sense, you just phosphorylate something, you get it activated, it just performs a function. At some point, there's a phosphatase that will sort of reverse this process and then inactivate the gap. So another mechanism to control the GTPases is binding to lipids. There's a series of gaps that can bind to different lipids. There's one of the best studied examples. It's a protein called chimerin. Chimerin has a domain called C1, and C1 is a domain that can bind a particular set of lipids called diacylglycerols, or DAG. Usually, as with the GEFs, the chimerin is in a sort of a tightly packed conformation in which the lipid binding domain cannot access to the lipids. And what happens is, when a receptor tyrosine kinases, it could be EGF, for example, is phosphorylated, it recruits a phospholipid lipase, which will cleave PIP2 into diacylglycerol. Diacylglycerol can now bind and recruit this protein from the cytosol to the membrane. This protein now opens up and gets activated, and it can now inactivate RAC. So this one is specific for RAC. And then other examples, protein-protein interactions domain. There's a Rho GAP called CD GAP. This is a protein that binds to another protein called intersectin. When it's bound, it's inactive. There's activation of the receptor of PDGF, the PDGF receptor. Somehow, I don't know how this happened, disrupts this interaction. When this interaction is disrupted, then the protein can get activated and again inactivate RAC. So these are only a few examples, but any combination of these three examples can also happen at the same time. You can have phosphorylation and then binding to lipid and then activation. You can have protein-protein interaction and then phosphorylation and then binding to lipids. These proteins have many domains, and usually people think that there's not a on and off switch, but more of a different layers of activation, like I showed you in the lab example. So if you, I'm sorry I should have mentioned this at the beginning, but if you want to stop me at any given time, it's okay. We can do the questions at the end, or we can do the questions at any time. If you have questions on a particular slide, just let me know and we'll go over it. So a lot of, so the most, I don't know what I put GEFs here, but mostly, so there's a couple of GEFs here, but there's some mutations of GEFs and GAPs that have been found associated with different diseases. There's not too many, but this list keeps growing over time. And one thing that I'll mention, and that I'm going to mention probably later too, is that there's hardly any mutation found in the GTPases themselves in disease. So there's not a disease that's associated with a mutation of a GTPases like you will see in RAS. So RAS is usually mutated in maybe 30–40% of all cancers. Rho proteins have not found, with a few exceptions, mutated in disease. However, Rho GAPs and Rho GAPs have been found mutated in a few diseases. For example, SOS, which is a RAS GEF, but also RAG GEF, have been found mutated in this disease. And what happened is this mutation promotes the loss of the autoinhibition. So again, it now has this protein constitutively active. This mutation now causes that this protein is always open, always in the open active conformation. It's always active, and that will promote, I guess, the constitutively inactive agent of RAC and also RAS. So FGD1, it's a protein that is found in this disease, Arscott syndrome. This is a disease that causes defects in the development of the skull and some skeletal structures. And we don't know exactly what it does, but the mutations are in the DH domain. So it could be either a change of specificity, it could be an inactivation of the exchange domain. Alpha peaks, which is another of the Rho GAPs, is RAC specific, has been linked to X-linked mental retardation. Again, we still don't know what this mutation. This is a truncation, so you lost part of this domain, the CH domain. It's still not known what's causing the disease. So a couple of other gaps, a CDC42 gap. Again, a gain of function. There's a mutation that causes this now gap to be constitutively active. It's now inactivating CDC42 continuously, and they've seen disruptions in the cytoskeletal structures, and then probably responsible for some of the defects observed. And then this gap also related to X-linked mental retardation. The mutation impairs the Rho binding site, so it's probably not active, but still maybe what happens when you render a protein inactive by mutation is you can still bind all the other components of the pathway. So where usually what happens is you're sequestering some of the upstream signaling, but there's no downstream effect. So usually these proteins can act as a dominant negative. It can block a signal that comes from the stream, and they don't transfuse any signal. All right, so I'll go a little bit over the regulation of Rho GDI. I showed these yesterday during my talk, so this is going to be a summary of what I showed yesterday. So if you have any questions from yesterday, feel free to ask them. I'm going to go over it again, and you'll see some of the same slides. So this is sort of the third layer of regulation. For many years, this aspect of Rho GTPase biology was sort of forgotten. Rho GDI was thought of more like a housekeeping gene, something that keeps the protein in the cytosol and inactivated, but not a whole lot more. I think that it's being recognized as a more active component of the Rho cycle now, and there's a lot more interest in trying to understand how it works. But essentially, as I showed yesterday, so I showed you that GEFs and GAP can be specific and can be very promiscuous. The Rho GDIs are very promiscuous. So the same Rho GDI binds to a lot of GTPases. So once you're bound to a Rho GDI, there's no interaction possible. So you cannot bind to effectors, you cannot bind to GEFs, and you cannot bind to GAPs. So the protein is essentially locked in a conformation. And to be able to activate it and to switch it to the active state, you need first to release it, and then a GEF or a GAP can act on it. And then, of course, I show you also that it's important to extract the protein from the membrane, and I'll show you that in the next slide, and also that it's sort of the equivalent of the total amount of the GTPases on the cell. I'll talk about it a little bit more in the next slide. All right, so one of the things that's important, and in part related to some of the work we did, is we think that the Rho GDI is important to deliver the Rho GTPases to the membrane. So that's one key function. And then once at the membrane, there needs to be an active event that displaces, that releases the GTPase from the Rho GDI. So once it gets to the membrane, it doesn't happen by itself. It needs to be actively modulated. It needs to be phosphorylated. It needs to be there needs to be some event. There needs to be protein-protein interaction. There needs to be something. So initially, a lot of people thought that this was going to be mediated by the GEFs, and it was very attractive. It was a very attractive hypothesis. You bring the protein to the membrane, the protein now can bind to the GEF and gets activated. It turns out that this was never observed. So even people try very hard to try to use the GEFs to displace the Rho GDI from the Rho GTPases. It doesn't happen. So it's something else. First it needs to be released, then the GEF needs to activate it. So once the GEF activated, it binds to the effector, performing function. One, it needs to be inactivated. It's inactivated by the GAPs. And then again, the other key function of the Rho GDI is to get it back to the cytosol. This again is an active recruitment. You need the GDI to get the GTPases out of the membrane, and what we've shown is that if you don't have it, then the protein stays in the membrane for a very long time. And then what we also shown is that if you don't have GDI in the system, then what happens is that this protein that's in the cytosol is very stable and very unstable, and it will go to the proteasome and get degraded. So the GDI is very important to maintain the pool of the GTPases in the cell. It functions sort of as a chaperone, preventing the misfolding or unfolding of the protein of the GTPases in the cell. Yep.
I have a question.
Yeah.
So, but all
you want
that.
To go what?
I'm sorry. Yeah. So all that process in terms of being released and going to the membrane and then being internalized again, all of that is happening within the vesicles, right?
No.
No, this is cytoplasm.
There's no vesicles here. So there's another component, and I'll show you. This is cytosolic. So if you fractionate membrane, and I showed a little bit of that yesterday, if you fractionate if you separate the membrane from the cytosol, GDI is always in the cytosol.
So it will get the Rho GTPase, which is a membrane protein, has lipids in it,
right.
And it will get it literally out of it.
Yeah, we'll just drag it out. And this is, in a way, a representation of how it happens. So the GDI combined to the Rho GTPases in two different places. This is sort of a weak interaction, and this little Pac-Man is the pocket that binds the lipid. So this weak interaction will allow then the lipid at some point to flip into the lipid binding pocket. And that is what allows the protein, even being lipidated, to exist in the cytosol. And that's all sort of what this structure shows, which I also showed you yesterday, is that if you look at the structure, you see that the lipid is really buried within the GDI molecule. Again, this is CDC42. Here's the nucleotide, and this is the C-terminus of GDI. You can see again the two sides of GDI that bind to CDC42. This part and this part, sort of what my cartoon looks like, two different sides of interaction, and then the lipid really buried into the molecule. So this is a very remarkable structure. And also, I showed you that some of the GTPases can be modified by two lipids because they have two cysteines. So these proteins cannot bind to Rho GDI. And the reason they can't is the two lipids don't fit in the pocket. It's just only one lipid that fits. So if you have two lipids, or even you have to artificially make them having two lipids, then the binding is interrupted. All right, so one of the things that I showed you yesterday, and I was going to just briefly mention today, is Rho GDI, in a way, determines the amount of Rho GTPases in the cell, because most of the GTPases are in the cytosol and bound to GDI, and if you don't have GDI, they get degraded. So in essence, the amount of GDI that you have in the cell will determine the amount of GTPases you have in the cell. If you artificially reduce the amount of Rho GDI, which I showed in the previous slide, all the GTPases get degraded. If you artificially increase the amount of Rho GDI in the cell, then over time, you'll have more GTPases. But that also means that there's a competition between the Rho GTPases that bind to the cell. And if you artificially increase the concentration of one, then over time you're going to compete with the other and decrease their concentration. And this is what I showed yesterday. If you express a GTPase, over time they will compete out for the other GTPases. They will be released from binding to GDI and enter the degradation pathway. And this is something that was very important because a lot of us, in many of our experiments, overexpress proteins. A lot of people have been overexpressing Rho GTPases to study their function. This is sort of an unintended consequence of overexpressing Rho GTPases. So one should be very careful when we use transfection or trying to overexpress a GTPase, in order to get to the level in which you decrease the concentration of the other endogenous GTPase.
Is the degradation instantly, or?
Well, it's hard to say. It's hard to say. So the way we do the experiments is you usually transfect GTPase and then you let it express for like 24 hours or 48 hours. I think that it's probably a process that takes several cycles. You have to compete with the GTPases that are bound, and you have to compete with the other GTPases that are being also synthesized at the same time of your overexpressed protein. So usually your overexpression is a lot more potent than the synthesis of the endogenous one. So I think that over time, it will depend on the amount of protein you synthesize, the amount of other proteins being synthesized, and the half-life of the protein, and all these kind of things. And then over time, you'll do it. We've never done a sort of a time course. And I guess that potentially you can do some sort of in vitro mechanism, in vitro system in which you may be able to determine that.
Does the competition varies with different GDIs?
That's a good question. So there's in essence there's one GDI in the cell. There's three GDI. GDI 1, 2, and 3. GDI 1 is expressed everywhere. Has very high affinity with the GTPases. GDI 2, it's a very poor binder to the GTPases. So the function is unclear. And then GDI 3 is tissue specific, and it's also very selective for the GTPases that it binds. It binds essentially to a protein called Rho G and maybe to another protein called Rho D, but it doesn't bind to the main one. But if you can flip it the other way, does the competition depend on which GTPase you're overexpressing? I think that it does. We don't have very hard evidence, but for example, we think that Rho A has a higher affinity for the GTPases than RAC. So then if you overexpress Rho A, the effect is more pronounced than if you overexpress RAC. You need higher amount of RAC. But this is all sort of circumstantial evidence. We don't have very hard data to show that, but we think that it may depend on which one. Yep.
When you activate a specific GTPase, like for example CDC42, then it goes to the membrane. The rest of the GTPases are sequestered then, or?
Well, in theory, yes, but since I've shown that only like 1 to 5% of the GTPases are activated at any given time, it's probably a negligible account. It's hard to detect. It should happen. If you have a free GDI, you're binding another one. But it also may be compensated by this same GDI extracting other proteins from the membrane. Because at any given time, you have an equilibrium between the amount that's being released and the amount that's being retrieved. So I think that probably at steady state, you have a balance. All right. So now that we're talking about that, the release. The release is something that is extremely interesting and something that people have not recognized for a long time. And the reason is very, very interesting is for these same reasons. The same GDI binds to multiple GTPase, and then it prevents any activity from happening to the GTPase that is bound. But the thing that we have to think is, like you said, when you activate one GTPase, I mean, the cell doesn't activate all the GTPases all at the same time at any given time. Usually you can treat a cell with a particular stimulus, and you know that you activate a particular GTPase. You activate Rho A, you can activate specifically RAC, you can activate specifically CC42. So how can the cell weaken the interaction specifically for just CDC42, specifically for RAC, specifically for Rho A? It's something that is still very poorly understood. The cell needs to be able to only release the GTPase that's going to be activated. And since the GDI binds to all of them, it cannot be a general mechanism that opens up the GDI and releases all of them, because otherwise all of them will be activated all the time. And that we know that doesn't happen. So there's some evidence that how can this happen. So, and there's a couple of different examples. So, and some of them are sort of more promiscuous, some of them are more specific. So there's one example showing that if you have a high concentration of phospholipids at a particular region of the membrane, then you are able to release specifically RAC1. So here when it says Rho, it's just the generic Rho GTPases. But so that's one example. You can have, for example, as I show in the example for chimerin, an enzyme that promotes the formation of PAG, an enzyme that promotes the formation of, in this case, it's phosphatidic acid. So you can have a high concentration of phosphatidic acid at a particular part of the cell. You can have things like lipid rafts that will recruit the Rho GDI bound to the GTPase, and then maybe that concentration of lipids will promote the prenylation, the prenyl group to move into the membrane and release from the GDI. So there's a couple of papers showing that this is possible. There are a couple of other papers that show, and when this paper came out, people started thinking about something that people call a guanine dissociation factor. People look for something that will just promote the dissociation, and they never found it. So this is the only example. So this is a protein this is a receptor that when bound, it binds to Rho GDI and promotes the release of the GTPase. So when this protein is bound to Rho GDI, then the Rho protein is released and it can get activated. There's a couple of other examples in which these proteins called ERM, esrin, radixin, and moesin. These are proteins very important for cytoskeleton regulation. When they bind, they can bind to Rho GDI, and again, they can promote the release. So this will be, I guess, the only two examples of something that could displace the Rho GDI by protein-protein interaction. But most of the examples that have been described have been, are related to phosphorylation events. And again, this was something that, that created a lot of excitement at the beginning because people thought, okay, if I phosphorylate a particular set of residues, I may be able to release a particular subset of GTPase. Maybe if I phosphorylate another subset of residues, then another subset of GTPases can be released. And there's some truth to that, but it's not the complete story. And there's a couple of examples here. The simple one is this one. You have a Rho GDI, for example, in complex with RAC here. If you phosphorylate in a particular set of residues, and usually the residues are adjacent or in proximity to the lipid binding domain. So what they're doing, these phosphorylation events are disturbing the binding to the lipid. When the binding to the lipid is weakened, now we can sort of flip and get into the membrane. So if you can phosphorylate different residues, promote the release and the activation. A couple of sort of more complex, sophisticated systems. In this example, PDGF promotes the recruitment of this complex, which is a diacylglycerol kinase, together with the RAC effector PAC, and RAC and GDI to the membrane. This enzyme will promote the formation of phosphatidic acid. Phosphatidic acid can activate PAC. PAC is a kinase, and then it will phosphorylate itself, and also Rho GDI, and then it can release RAC1. So this is probably one of the best studied examples, but I think that more examples are going to be described in the future, in which, again, there's a combination of recruitment, protein-protein interaction, phosphorylation, and release. So here's a summary of all the, it's a little bit cut, but I'll guide you through it. So this is an example of all the phosphorylation events that have been described so far. So in the top part, you don't see it here, but these are all serine phosphorylations in the amino acid serine by serine threonine kinases. So threonine here, and then these three in the bottom are tyrosine, so tyrosine phosphorylation. And then There's another event of phosphorylation that I didn't mention that is also very important, is the phosphorylation of the Rho GTPases itself. Some GTPases can be phosphorylated, and this generates the opposite effect. When the GTPases has phosphorylated, the affinity for the GDI increases. So this is a way that the cell has to terminate the cycle. When the phosphorylated are, when the GTPase has phosphorylated, then they bind with high affinity with the GDI, they get extracted from the membrane and the activity goes down. And this has been documented in several cases. Not all the GTPases can get phosphorylated. For example, RAC does not have this residue, but Rho ACE and CC42 do. In particular, this one, serine 188, is one that has been very well studied. There's another sort of pattern that we can just visualize when looking at this table. If you look at all the serine phosphorylations, I think that I can, yeah. If you look at all the serine phosphorylations, if you look at the effect, most of the phosphorylations in serine promote the dissociation of a specific GTPase or a specific subset. For example, phosphorylation in serine 34 and usually the phosphorylation in tandem, 34 and 96, promotes the release of Rho A, or it was described in one case of Rho G. Phosphorylation of serine 101 and 174 promotes the dissociation of RAC, and maybe to a lesser extent CDC42. So it seems that phosphorylation in serines confers a little bit of specificity in the release. However, when you phosphorylate tyrosine, and this is these three ones at the bottom, what you can see is that in most cases it's Rho RAC and CDC42. So you would release everything. So a phosphorylation in tyrosine, it's a lot more sort of drastic event and affects the affinity for the Rho GTPases in a more, I guess, dramatic way, in which you just release all the GTPases. Serine, in contrast, applies, there's some specificity that can be conferred by phosphorylating serine. And then, as I showed you before, if you phosphorylate the Rho GTPase, then you increase the affinity. So again, there's always a balance of what you can do. You can phosphorylate the Rho GDI to release the GTPases. You can phosphorylate the GTPases to increase the affinity. All right, let's talk about Rho effectors. Again, as Rho GAPs, the Rho effectors, there's a lot known about a few of them, and there's almost nothing known about all the rest. So there's a few of them that have been extensively studied, and then there's the rest of them that are completely forgotten. So Rho effectors comprise a big family. So as of today, more than 100 effectors have been identified. So I show you in the first few slides, the effectors bind to different residues. Mostly in these two regions is the GTPases, switch 1 and switch 2. And in some cases, there's some other residues that can contribute to the binding. There's always a little bit of difference between one effector and the other, but for the most part, the regions that are important for binding seem to be the same. There's a lot of kinases within the effector family, but then there's also scaffolding proteins that don't have any particular activity, no catalytic domain, and mostly a collection of protein-protein interaction domains. So this is just a brief summary of some of the very well-known effectors. The two best characterized effectors for Rho A are Rho kinase. This is a serine 3 amine kinase that promotes the formation of stress fibers and induces contractility. So when you saw that picture in which the stress fibers are induced, it's mostly a Rho kinase effect. So Rho kinase will phosphorylate certain components, and I'm going to show the pathway in a few slides later. And it promotes contractility, formation of stress fiber, actin bundling, and contractility. The second best known effector for Rho A, they're called diaphenos or DIA. These are proteins that promote actin polymerization, but also polymerization of a particular type of actin, which is linear actin filament. So for RAC, so the best characterized effectors are PAC. PAC are against certain threonine kinases. They are mostly involved in the polymerization of actin at the edge of the cell and also in transcription through this JAK kinase pathway. Then, of course, for CDC42 and for RAC, these two proteins that perform similar function are the ones that are involved in formation in what's called branched actin. So you can polymerize actin in two different structures. You can form linear filaments of actin, or you can form actin filaments that are polymerized in what's called branch, and they form an angle which is at 70 degrees. This is what usually happens at the edge of the cell when the cell is migration, and the very leading edge of the cell. You have this network of branch actin, and these two proteins, WASP and WAVE, which are RAC and CC42 effectors, are the protein. This protein form a complex. This complex recruit a complex of proteins called ARP23. This is a protein that basically creates this 70-degree angle. It's an actin nucleator, and it will basically align the actin over a previous existing filament in a 70 degrees. And through many of these events, you form this branch actin. And then some other effectors. As you can see, most of them are either serine threonine kinase, scaffolding proteins, and then some others are lipid kinases. The way they bind to Rho A and RAC is most of the times to very defined domains. The ones that bind to RAC or CDC42 are called CRIB, CDC, RAC, C42 RAC interactive binding domain, and then REM is the Rho effector motif, and then this is the Rho kinase homology domain. So they have slightly different domains, and these are the domains that we usually use in some of the assays that we use to pull down the active polar proteins. So we just basically cut a piece of this protein that can bind specifically to the active form of the proteins, and we use them to pull down the different GTPases. So again, these proteins can only bind the active form of the GTPase. All right, so how these effectors are regulated? The most common mechanism is the same mechanism that you will see with Rho GAPs. These proteins are sort of folded over themselves. They're inactive. They're in a conformational inactive state. Maybe the effector domain is binding another domain within the same protein. Once the GTPase gets activated, it will bind to the effector domain, open up the molecule, and whether it's a kinase or something else, it will transduce a signal downstream. In some cases, this signal can be also reinforced by other proteins or lipids, for example, binding other cofactors, binding to the open molecule. And that can happen, for example, when a cofactor binds to one of the domains that is now exposed. And it could be, for example, PIP2, or it could be a different lipid. It could be also another protein. So there could be sort of two signal intensities depending on the initial stage and the late stage of the signal pathway. In some cases, the effector forms part of a complex that it's inactive, and what binding to the GTPase does is to release the effector now to the cytosol, and now it can just promote the signal once it's being released from this sort of inactive complex. So it's sort of a negative way of regulation. Then also you can have the opposite effect. You can have an effector that is always active, and when it's bound to the GTPase, it's now inactive. So you can have a sort of negative signal by binding to an effector. So I'm going to go through a few of the pathways that have been described for the Rho GTPases. You don't need to get too worried about the details. These are just examples. The pathways are extremely complicated, and this is a very simplistic view of them. So don't worry too much if you don't remember or if you don't know the name. This is just to give you a flavor of what's known about basically a typical example of a Rho A pathway, a RAC pathway, and a CDC42 pathway. So as I told you in the past few slides, Rho kinase and MDR, so this protein called diaphanous, are two of the best known effectors for Rho kinase. In the end, what they will do is to promote adhesion, contractility, and the formation of stress fibers. So the way they do it is both by affecting actin polymerization and also the activation of myosin, which is what will promote the contraction of actin. So Rho kinase is a pyrimidine kinase, as I showed you before, and then for this diaphanous is a part of a big family of proteins called formins. So Rho kinase is a kinase. It will phosphorylate different substrates. A couple of substrates are limb kinase, another kinase. This is the myosin light chain. The myosin light chain, that when it gets phosphorylated, it's active. And Rho kinase, it will also phosphorylate and inactivate myosin light chain phosphatase. So there's two ways of increasing the phosphorylation state of myosin by phosphorylating it directly and inhibiting the dephosphorylation state. So it's a sort of two-way. Rho kinase has a backup system to ensure that the myosin light chain gets phosphorylated. So once a limb kinase is activated, it phosphorylates another protein. This is an active, also an actin binding protein called cofilin. This protein promotes the depolymerization of actin. So when it's phosphorylated, it's now inactive. So this is sort of an inhibition pathway that promotes stability of the actin filament. It prevents the actin filament from depolymerization. So phosphorylation of myosin light chains will increase myosin activity, so that increases contractility. The activation of these proteins, these forming proteins, will promote the formation of actin filaments, and altogether this will promote the formation of stress fiber, contractility, and will promote the formation of focal adhesions, and also stress fibers and contractility. So RAC is also very complicated, but I guide you through it. So the three, again, main effectors that I'm going to talk about. Actually, it's two if you count wave as one, I guess. So the scaffolding proteins and the kinases. So there's always some sort of a pattern here. You're always activating a kinase, a scaffolding protein. So the scaffolding proteins are these proteins that I told you about, wave and wasp. Wave and wasp are actin nucleators. What they will do is promote the growth of actin filaments in this particular angle. So you form these branch actin filaments. Wave and wasp exist in a complex, two different complexes that are inhibited. And what happens when RAC binds to these proteins, they release wave from the complex and it activates it. And now wave can promote actin polymerization and the formation of different structures in the cell, mostly protrusions at the edge of the cell called lamellipodia. So PAC, the serine threonine kinase, will sort of have the opposite effect of what I told you on Rho. PAC will still stabilize the actin by phosphorylated limb kinase. So the same as Rho kinase will phosphorylate limb kinase, promote the phosphorylation of cofilin, which now will be inhibited and just promote the stabilization of the actin filament. But in the case of the myosin system, PAC will inhibit myosin light chain kinase, so myosin light chain phosphorylation will decrease, and then it will also inhibit myosin, it will phosphorylate and inhibit myosin heavy chains. So the overall activity of myosin will be lower now when RAC is activated. So the stress fibers will decrease. And this is a common theme that you'll see over and over again if you read the Rho literature. Rho and RAC are antagonistic. They don't like each other. So if you have high Rho, you usually have low RAC and vice versa. So a lot of the things that one signals through are opposed to the other. And I'll show you a few more examples. So CDC42. Okay, so let's start from the bottom up. Most of what CDC42 does is to promote cell polarization in many different forms. Again, I told you at the beginning, axon and neurite outgrowth, polarization of epithelial cell, even polarization of a migrating cell. When you have a migrating cell migrating in a particular direction, many of the things need to be aligned in the direction of migration, including the nucleus and the Golgi and some of the organelles of the cell need to be pointing in the right direction. And when the cell turns, all these organelles get reoriented. This is all CC42. Golgi reorientation, centrosome reorientation, the polarization of microtubules, actin polymerization. All of these things are important. So this CC42 is the master regulation of polarization. How does it do it? Part of it is through the formation of branch actin, and I show you wave is very similar to wave, but with CDC42. It activates this protein called R23, which promotes phosphorylation, promotes formation of actin. CDC42 plays also a big role in polarization of microtubules, and they do that by the function of PAC, which is again is a serine threonine kinase, and through the proteins, microtubule binding protein called STAT-MIN. Again, the same effect of RAC, stabilization of the actin filaments by inhibiting co-filling. And then part of what determines the polarization is this complex of proteins called PAR6. These are proteins that are important for formation of cell-cell junctions. So when epithelial cells polarize, they just bind, they form adhesions, they adhere to each other, and they form tight junctions and adhering junctions. And that's important to maintain the polarized phenotype. This is part of what makes it work. And then some other things like membrane traffic is important also for the polarization of the cell, because when the cell is polarizing, some vesicles go to the apical surface, some vesicles go to the basolateral surface. So all of this is controlled by CDC42. And again, so there's a kinase, a scaffold, and there's also tyrosine kinase in the case of CDC42. So again, don't worry too much about the details, but the point I want to make here is all these pathways are occurring in the cell simultaneously at any given time. So this is an incredibly complex system, and it needs to be regulated both temporally and spatially. So, and I put here the example of a migrating cell. And again, you can see how the phenotype, the polarization phenotype. You have a leading edge where you have branch actin at the leading edge. You have the activity of RAC and CDC42 at the edge, and then at the middle and the back of the cell, you have formation of stress fiber. You have the formation of focal adhesions. You have contractility. You have events that promote the retraction of the tail in the direction of migration. All these things have to occur at the same time. You need to move the nuclei too, something that we don't think about too much, but the nucleus is big and heavy. When the cell moves, it also needs to be transported with the cell. So there's all these pathways occurring simultaneously in the cell. So there has to be a lot of compartmentalization. There has to be a lot of segregation of the signaling pathway. It's not all just floating in the cytosol, just mixed together. It's just very, very compartmentalized. And there's a big challenge to try to understand how this works. So I'll show you a couple of movies just because I like them a lot, and also it will illustrate what's happening in the cell. So, and what different things you expect to see in the cell. So at the very edge of the cell, I told you that you are forming this branch actin. And this branch actin is also promoting the formation of these small adhesions. So this is what the cell is starting to attach to the substrate. So these are very small adhesions that are very unstable. Once these adhesions form, they get stabilized and they start growing. And once they grow, they promote the formation of stress fibers. And the stress fibers will just generate tension, and this tension again will promote the maturation into what's called focal adhesions, and then this focal adhesion eventually evolve into longer structures that are called fibrillar adhesions. So what you see here is very small adhesions that form and disappear. What you're going to see here is very small adhesions that some of them will develop into bigger one. And then here you'll see that in the middle of the cell, you'll see bigger adhesions that are more stable and they don't turn over. And this is just by looking at a fluorescent protein that's part of these focal adhesion complexes. So what you're imaging is a fluorescent marker of focal adhesions. You'll see this one. Lots of events, so little things that just form and disappear. And nothing is very stable. Everything sort of turns over very fast. This one, you see that the cell is growing now. And some of them, like this one here, start growing and maturing to focal adhesion. So only a few of them do, but they do it very nicely. And then this one is a similar one, just lower magnification. So you can see what happens in the middle of the cell. Now, everything that's in the middle of the cell is now a little more stable. It just stays and it doesn't move too much. What's at the edge of the cell is what's very dynamic. So these things induce an incredible amount of signaling. So what's happening there is just promoting phosphorylation, polymerization, contractility. And there's hundreds and hundreds of proteins that are being recruited there, performing different functions. And again, I can try to put it into context with one of the players that we saw. So most of these things are mediated by the function of these receptors called integrins. And the integrins are the receptors, the transmembrane proteins that can bind to the extracellular matrix. Extracellular matrix being proteins like fibronectin, collagen, things that are in the connective tissues, in the basement membranes, in the interstitial parts of the tissues. So this is a very important part of most of our tissues. So the cells attached to the intracellular matrix, and not only that will confer mechanical integrity, but it will also promote an incredible amount of signaling. So if you see so this is what will happen at the very edge of the cell. The integrins will bind to the extracellular matrix. They will activate a series of kinases. These kinases will activate some different signaling components, including exchange factors. These exchange factors will activate RAC and CC42, which in turn activate actin polymerization machinery, which will induce the formation of the leading edge of the cell. So again, what you have is signaling components that will activate GEFs, GTPases, and effectors, which is what we've been talking about today. So what happens in the middle of the cell? In the middle of the cell now, this part is not active anymore. So now what you have is similar components. Again, kinases will activate some lipid kinases. Lipids can activate exchange factors. We have some exchange factors that can activate Rho A. Rho A will activate Rho kinase and the formins, as I showed you before. This will promote polymerization of actin, promotes also contractility by activating myosin, and this is what causes these focal adhesions that are very unstable to stabilize and grow. And this will promote strong adhesion to the matrix. So again, you have GEFs, you have some GTPases, and you have some effectors. So then, let me go back to that. So then in the tail, you need to, so what happens at the back of the cell is that you need to now release this adhesion. So initially you have very unstable adhesion, then you have stronger adhesion. Then at the end, at some point you have to bring the tail forward. So you need to release the adhesions. There's a couple of different ways to do it. There's a way to do it by phosphorylation, some of the phosphorylated components by this phosphatase called P10. But there's also sometimes what the cell does is just releases those adhesions. It leaves them behind. So what you see is that the cell at the end of the cell is just losing little pieces. And there's also another way to do it is using proteases. The protease can specifically cleave some of these signaling components, just inhibiting the signaling pathway. So, okay, I have a few more minutes. So, I just have introduced you a little bit of what happens between Rho and RAC. Rho and RAC don't like each other. They really go in different directions. And most of the GTPases will signal through a particular pathway, but will also influence the activity of other GTPases. And this is a very common thing And something that also needs to be taken into account when you're looking at a particular GTPase, it's probably a good idea to look at the activity of the others, because they're always connected. And there's many different ways that have been described these GTPases are connected. And in the most simplistic way of looking at it is one GTPase can inhibit the activity of the other GTPase. So that's one way it could happen. The other way is one GTPase can induce the activation of another GTPase. So it could be positive and negative feedbacks. And there's examples for both of them. So I'll start making things a little bit more complicated. So we'll go with the inhibition. How can you inhibit the activity of another GTPase? So let's say Rho or GTPase 1 gets activated. What can happen is that the activation of this GTPase can inhibit the activity of a Rho GAP for the second GTPase. So you activate, for example, RAC, you can inhibit a Rho GAP, now Rho gets inactivated. The other way you can do is by activating a Rho GAP. So by activating a gap, you can inhibit the next GTPase. So you activate a gap of a certain specificity. There's also a way you can do it through Rho GDI. You can either you can compete out the other GTPase, you can promote the phosphorylation of Rho GDI, as I showed you before. And then you can directly influence some of the downstream effector or targets of the signaling pathway. For example, RAC can inhibit some of the effectors that Rho can also activate, so preventing Rho signal. And there's this positive signal. I guess you can think about this in sort of the opposite direction. A Rho GTPase can activate a GEF for the other GTPase, so one promotes the activation. The opposite would be to inhibit a gap, and the same for the opposite effect of the GDI and the effectors. So most of these examples have been found and described. So this is an incredibly complicated picture. So this is just one summary of what happens between RAC and Rho. I show you that RAC promotes the formation of lamellipodia, these unstable nascent adhesions, and promotes migration, while Rho promotes stress fiber formation, promotes stable adhesions, and so another different type of migration, and this I didn't talk about. But how can Rho influence RAC and vice versa? For example, Rho A can activate Rho kinase. What Rho kinase does is prevents this RAC GEF to just be recruited to the membrane, so it just prevents RAC from being activated. It also phosphorylates and activates RAC gaps, so it inhibits RAC activation. And then in some other cases, Rho A can also activate RAC activity, and this is sort of a different example in which Rho activates DIA and DIA activates RAC. So how does RAC do it? RAC can also activate Rho A in some cases by this exchange factor, but also can activate Rho GAPs inhibiting Rho A activity, and through PAC phosphorylates a series of Rho GAPs and inactivates them, and then preventing Rho A activation. So there's an incredible amount of communication between the Rho GTPase. And one shouldn't think about this pathway as being isolated in this. When you're affecting one, you're probably affecting many others by just these crosstalks. And this is just a different way to look at it, but some of the same things that I've already shown. Some of the data that people have collected over the years show that there's a very restricted Rho A activation at the leading edge, and then there's some RAC activation in the sort of in the middle part of the lamellipodia, and then there's Rho activation at the back. And this can be controlled by some of these different crosstalk pathways that I show you. And I'm going to skip that because I want to show you before we finish how we study the GTPases. So through the years, people have developed different assays to study Rho GTPases. And ideally, what we want to see is when and where and how much they get activated and how they're being regulated. So we use a series of different assays. So the first one described is what's called a GTPase pull-down. And in this assay, and I'm going to talk a little bit about them in detail. In this assay, what we do is we have a protein that we purify in bacteria, and it binds specifically to GTP, to the GTP bound form of the GTPases, so we can analyze in lysates how much of the particular GTPase is active at any given time. We can also use a similar technique in which we use mutations of the GTPases that are in this nucleotide-free state, and we can use them to pull down GEFs, and we can use a similar mutant that is constitutively active to pull down GAPs. So again, the end result of these experiments, we can analyze how much GTPases, how much of a particular GEF, how much of a particular GAP we have active in the cell. We can also try to analyze enzymatically how these GEFs and GAPs work. And this is a way in which we can test both specificity and efficiency of a particular GEF and a particular GAP in vitro. And this, again, what we do is we have purified GEFs or purified GAPs, and we have different fluorometric assays in which we can measure either the exchange of a nucleotide or the hydrolysis of the nucleotide, depending if we're looking at GEF or GAPs. And then the latest tools in the GTPase toolbox has been the development of what's called biosensors, and this was pioneered by the lab of Klaus Hahn and some other labs. And what these tools do is they allow you to visualize in live cells where and when these GTPases are activated. And I'll show you some examples of that. Okay, so the first assay, how to analyze Rho GTPases activity. What we use is GST fusion protein, which we express in bacteria, fused to an effector binding domain. If we want to study Rho, we use an effector that binds specifically to Rho GTP. If we want to study RAC, we use a RAC binding domain. And then this is what we usually look at. We break open the cell. If we want to try different conditions, we have different conditions. We incubate the lysate with the effector binding domain fused to GST, and then we run them in a gel and we blood both for the GTPase of interest. And this is an example in which the cells were plated on fibronectin, which is an extracellular matrix protein at different time points. And what you see is that the activity of Rho, this is the active pool of Rho A, this is the total pool of Rho A. And what you see is that the Rho A activity goes down in the first 20 minutes. And this corresponds with a high RAC activity, what I show you when the cells started forming these nascent adhesions. Then later in time, when focal adhesions start to mature, you see a corresponding increase in Rho A activity. All right, so what are the advantages of this assay? This assay is actually a really nice assay, but it has a few disadvantages. One of the main disadvantages is that you're only seeing the average activity of a particular GTPase in the cell. If you, for example, have a high Rho at the front and low Rho in the back, your average may give that the Rho GTPase activity doesn't change, because you're just measuring the whole activity in the whole cell. So that means that you don't have any spatial resolution. You don't know where you are getting the activation. And then the other drawback is that it requires a large amount of protein. You need to use a lot of cells. If you have a very precious sample on a very small amount of material, it's very hard to do this assay. So this has been partially solved by another assay that uses an ELISA version of this. You can use a lot less protein in this assay. So it's potentially can be resolved by using this other assay. So the GEF assay, as I show you, you use a mutant that mimics the intermediate state in the exchange reaction. So in a similar way, you use a GST fused to this Rho binding, to this Rho GTPase of interest that cannot bind any nucleotide. This will bind with high affinity with the active pool of GEFs, and then you can see them in a gel. And this is just an example in which we have a GEF, an effector, and a GAP, and with the nucleotide-free version, we only bind to the exchange factor, but not to the Rho GEF, not to the effectors, not to the GAPs. So if we use a gap, so to pull down gaps with a GTP bound form, constitutively active, and this is again the glutamine that I show you in the structure that is very important for the exchange. We're mutating the glutamine for leucine. Now it cannot be hydrolyzed, and it binds to the gap and it remains bound, so we can bind, with this mutant we can bind both gaps and effectors, but not GEFs. And then the in vitro, I'm just going to show you the in vitro GEF assay. In this exchange assay, we're measuring the speed and also the specificity of an exchange reaction, so a particular GEF of interest. And the way you do is you have your GEF together with GTPase, and you use a fluorescent nucleotide instead of GTP. This fluorescent nucleotide changes its fluorescent emission once it's bound to a protein, so it increases. So when it's incorporated through the exchange reaction, now the fluorescence increases. So in this assay, what you can see is an exchange factor for CDC42 called DIPS. So when you have only CDC42, the release is very, very slow, and the fluorescence increases very slowly. At this point, what you do is you add to this assay the purified exchange factor. And in the presence of this fluorescent nucleotide, the fluorescence increases significantly, extremely fast. So this is an exchange factor that works very well for CDC42. If you do the same for Rho A, this is also exchange for Rho A, and then again you have Rho A only. When you add the exchange factor, it exchanges. And this is a very poor exchanger of RAC. And you can see RAC only, when you add the exchange factor, it doesn't do as well as the two others. So this is a way that you can say, okay, this GEF is really good for RAC, for CC42 and Rho, but not for RAC. All right, let's talk about biosensors, and then I think this is my last few sets of slides. Just to talk about biosensor, I just need to give you a very brief introduction of FRET. You probably heard about FRET, and this is fluorescence resonance energy transfer. This is a case in which you have two different fluorescent proteins that, when they get into close proximity, by exciting one, you can transfer some of this energy to the second one and induce the emission of the other one. So this fluorophore will be excited at this wavelength and emit at this wavelength when it's not in close contact with this protein, with this other fluorophore. If the two proteins interact and you get them in very close proximity, then you get emission of the second fluorophore. And there's an equation that you can use. The proportion of this emission is related to, inversely correlated to the square of the distance, so you can make calculations numerically of how much emission and what distance and these kind of things. So most of the biosensors used to study GTPases will use some kind of FRET analysis. So I'll show you a few of them. The simple one, the most simple way to think about a biosensor is, can I find something that binds specifically to the active portion of the GTPase and I can detect it in the cell? So that's a biosensor. So the simple one will be to do the equivalent of the Rho GTPase assay in a cell. So to express an effector binding domain, fuse to a fluorescent protein, and look if it changed localization when your particular GTPase of interest is activated. And I'll show you a really, really nice example. So this works, but it works only in a certain system. This lab in Wisconsin, Wilbur-Ment, they've been using this in whole Xenopus embryo. And what they do is they create a wound. They just hit the embryo with a laser, and it creates a little bit of hole. And then they look at what happens to Rho A activity and CDC42 activity. And they do that by having the Rho binding domain fused to GFP and the CDC42 binding domain fused to a red fluorescent protein. And this is quite striking. You're going to see Rho A activity in red, CDC42 activity in green. When you create the wound, and then it's amazing how they get compartmentalized. You see Rho activity at the very edge of the wound, and then behind Rho A, you get CDC42 activation. So this is extremely beautiful and elegant imaging, but this system doesn't work in most systems. And the reason it doesn't work is because by overexpressing these proteins, you're basically acting as a dominant negative. And I think that the reason it works in whole embryos is because these cells are very big and this gets diluted and the concentration, and there's many reasons that this may work in this system. But it usually doesn't work in a cell culture system. So again, this could act as dominant negative, but it will allow you to measure multiple GTPases, and one of the advantages is that you're looking at the endogenous GTPases, so you're not overexpressing GTPases. So the next, I guess the next sort of iteration of this system, and people have been improving in the development of biosensors over the years, is to make a sort of a thread-based sort of improvement of this, in which you have an RBD binding domain or a PBD, a RAG binding domain, bound to two fluorescent molecules. So actually you have a PBD, no, this one is different, sorry. So you have, in this case, a RAG binding molecule in which you cross-link to a fluorescent molecule. And then you express it together with a GFP form of RAG or CC43. And again, when they bind these two together, when it's active, it will fret, and you can detect the fret. This was a very early development. It was a very poor fret, and it also required both overexpression and microinjection. So this was a very difficult system. So it's not being used anymore. So I guess the next improvement was to try to put everything in one single molecule, in which you have both, for example, Rho A fused to a fluorescent molecule, fused to the effector binding domain and another fluorescent molecule. The principle is when Rho gets activated, it's going to bind this domain. This will going to come in close contact, and it will give you a Fred signal. This design is sort of the early development of these sensors, and you can notice that it has the lipid domain in the CFP and not in the Rho A. And I told you that the lipid domain was very important for